So I'm going to talk today about advanced methods in BioSAXS. If you haven't had a chance to look at the previous webinar, I'll briefly summarize it. Last time I talked about just the basic theory in SAXS data collection, where the scattering actually comes from, what types of experimental protocols you want to use, and additionally, one of the important things is that you want to make sure that your data is of the most pristine quality that you can get. You want to make sure you have non-aggregated, monodispersed solutions that are not interacting with itself. And the reason for that is because once you have a very high quality SAXS profile, you can begin to use some of these more advanced methods to really get at additional information within those curves by combining other methods with it. So some of the methods that I'm going to go through today, I'm going to start off with where I kind of left off with the last session, which is envelope reconstruction. Then I'm going to talk about how you can utilize high-resolution structures and combine that with SAXS data to do rigid body modeling and docking and flexible fitting. And then I'm going to talk about some dynamic information that you can get through Ensemble modeling and continue with mixtures, combining all the information from several different techniques using hybrid modeling, and then talk about contrast matching and time-resolved SAXS. So the first series of methods here is mostly going to be focused on software developments that have been able to take place with the advances in computational power over the last few decades. And it's really going to be focused on what you can do with a conventional SAXS profile and really extract as much information as you can out of it by adding as many constraints and as much a priori knowledge as you have about your system. I'm also going to talk about some other methods, contrast matching and time-resolved SAXS, that are less conventional types of scattering that can also be able to probe some different types of information. So one of the important things from all of these different methods that I'm going to be talking about, they all use different ways of creating a model and comparing it with your SAXS data to determine whether or not that model is accurate. So they all require the accurate calculation of the reciprocal space scattering profile from your model. And one of the important things from this is that it's not important that it's just accurate. It also needs to be computationally fast, because several of these methods require the evaluation of thousands or even millions of different possible candidate models. And so these algorithms need to be very fast and be very accurate. Many different algorithms have been produced for this exact purpose, and they're primarily based on two different ways of calculating the scattering profile. The first is the Debye equation, which is shown here, and another is spherical harmonic expansion. So just to give you a quick example, the way they calculate is you take your model with atoms, n atoms, and for each atom pair I and j, you calculate the form factor of that particular atom as a function of Q, and then you calculate the form factor of the partner atom as a function of Q, and you multiply it by this sync term here where it includes the distance between the atoms I and j. And you do a double sum over all atoms j and all atoms I. And so you can see, because it's a double summation, it can actually take quite a while to calculate it. It goes as n squared. So if you have a very large object, it's going to take a while to calculate these. So people have devised different methods to be able to calculate these efficiently. Now, one of the additional things that needs to be taken into account is modeling the solvents. So when you actually do a SAXS experiment, I described in the previous talk that you actually are collecting data from two different samples. One is going to be your protein sample, and the other is going to be a buffer blank. Now, there's two reasons that you collect the data from the buffer blank. One is that you need to correct for any kind of capillary scattering or air scattering or anything like that that's going to contaminate your signal. The other reason is that the bulk solvent itself is going to have scattering. It's going to be some kind of average background. And so you need to subtract that out of your data. But while that subtracts out the bulk solvent scattering, it doesn't subtract out the scattering of the contribution of a solvent shell around proteins or biomolecules. And it also doesn't take into account the excluded salt volume within the protein molecule. Now, the bulk solvent is going to have one average electron density for, it's mostly water, so it's going to be around 0.33 electrons per angstrom cubed. But what they found is that the solvent shell around within a molecule generally is a slightly higher density, maybe by about 10 or 15%. And this slightly higher electron density will have a different contrast with the surrounding region, and so it will contribute slightly differently to the scattering of from your experiment. Now, importantly, the solvent contribution will actually vary from molecule to molecule. The surface properties of that molecule will be based on electrostatic interactions, hydrophobic and hydrophilic interfaces. And so there's a heterogeneous distribution of the solvent around the molecule. And some programs take that heterogeneity into account and others don't. It depends on what your particular goal is for that experiment. Additionally, different chemical conditions can affect the scattering terms. If you have a high salt, or if you have some kind of high electron density component within your solvent, you'll need to model that accurately in order to be able to account for that contrast. Some of the methods use coarse graining in order to speed up the computational cost. And generally, because SAXS is low resolution, these coarse grain methods are quite accurate. They're almost as accurate as all atom methods, but significantly faster. Some of the most common algorithms are probably Chrysol, I would say, is probably the most common, and FOXS is also very common. But many others exist, and several of the modeling programs that I'll be talking about have their own built-in solvent and scattering profile calculators, but they generally all have a trade-off between accuracy and speed. And just to give you a quick example, here's a table from a recent review that talks about some of the different programs that exist using different methods for calculating the SAXS profile. And they use either spherical harmonic expansion. You notice that there's a lot that use the Debye formula. They have different ways of modeling the solvent, either through an implicit solvent shell or explicitly, which allows for this heterogeneity within a solvent distribution. And several of them have online servers as well as downloadable profile calculators. This is just a quick example to show you that generally they're pretty accurate. As you can see, that they use the solvent as a fitting parameter, and they all will fit it slightly differently. I think some of them now have the ability to, when they're modeling the solvent, they actually won't use it as a fitting parameter. They'll explicitly calculate it, and it ends up being quite accurate. But those generally take a lot longer to calculate. So this is just a quick comparison of some of these methods. So I'm going to talk first about, in terms of modeling software, about envelope reconstruction. So envelope reconstruction is probably one of the more well-known attributes of SAXS. And envelope reconstruction involves creating a three-dimensional ab initio envelope to model the one-dimensional profile that you get from your data, from your experiment. Several different programs exist for this, maybe not several, but different programs exist for this. The most common is probably DAMIN and DAMIF. DAMIF is just a very fast version of DAMIN. Now, basically the way these programs work is through what's called bead modeling. And so what you start with is a large grid of beads that are either hexagonally packed or packed in some way, and you essentially sample lots of different conformations, different permutations of the bead array. The problem with this is that if you say you have 1,000 beads, in order to calculate all the different types of models that you can generate from that grid of beads, you end up with 2 to the n, or 2 to the 1,000. And this is too many different conformations to be computationally feasible. And so different approaches, such as Monte Carlo-like approaches, are used to try and find a minimum within that large sample space without having to sample all of it. Now, one of the problems with these direct Monte Carlo methods is that they can fall into local minima pretty easily, and so people develop simulated annealing algorithms to try and find a global minimum using random seed generation. One of the important things about envelope reconstruction is that you want to avoid overinterpretation of envelopes. So SAXS is one of these methods that no matter what data you give it, it will always produce an answer. Regardless of the quality of the data you give it, it's going to give you some answer. And it's unlike crystallography, where sometimes crystallography, you won't get an answer. Your molecular replacement will fail, and that something won't work, and you won't get electron density or something along those lines. But SAXS, you'll pretty much always get an answer no matter what you give it. So you want to make sure to avoid overinterpretation of these envelopes. So as I mentioned, DAMF uses a dummy atom bead modeling approach. And in order to be able to model a protein accurately, what they do is these programs use physical constraints and add them as penalties into a scoring function. So that way you can ensure that you're getting a model, an envelope that actually looks like a protein. You know that you're not going to have some random bead sitting over here and the rest of your protein over here, because proteins are connected objects. And so it uses penalties like looseness and compactness, and adds them as a separate term within the scoring function, so that you're simultaneously looking for the best fit to the data and minimizing these penalties. And this is kind of akin to what you can think of as restrained refinement in crystallography. If you're familiar with crystallography, you add in ancillary information such as chemical bond distances and Ramachandran angles, knowing that these are well verified parameters, or rather observations that you can add to your as a constraint within your modeling. So DAMF uses the spherical harmonic approach to calculate the scattering of a model. The contribution from the solvent shell is approximated implicitly, and sometimes when you get out to higher resolutions, this can lead to some errors. So if you're running ab initio envelope calculation using DAMF, they generally recommend, the people that developed it recommend that you limit your data to lower resolution data where S or Q times RG is around 7 to 8. If you do want to use that higher resolution information, you can use a program called GAS4, and this uses the Debye approach to calculate the scattering and uses dummy residues that are connected laterally to each other rather than the grid of dummy beads. Additionally, it also models the solvent explicitly, and when you combine all this, this is why they say that you can use this for higher resolution data. So when you're running envelopes, I mentioned that you have a simulated annealing protocol that has a random seed. So if you were to run the same exact data 10 times, you'd get actually 10 slightly different envelopes. And the idea is that because the random seed is slightly different, these bead configurations are going to be slightly different. So what you want to do is form what's called a consensus envelope by calculating, say, 10 different ab initio envelopes and averaging them. And you notice that most of the envelopes are, the general shape is the same, but you notice there's beads in certain locations that are not in other locations in other models. And so by building up a consensus envelope, what you're trying to do is locate which beads are actually from the signal in the data and which beads are just a consequence of the artifacts of the random seed and the noise in the data. Another way to approach this is through clustering. This is a more recent development within the last few years, and clustering allows you to, instead of building up a single consensus envelope, you can actually find clusters within your pool of ab initio envelopes, and that allows you to kind of assess or access some of the different ambiguities that might be present in some of your envelope reconstructions. It can sometimes give you information about dynamics or different conformations that might be present, and so clustering can be another approach. So that's ab initio envelope modeling, and so that uses essentially just your scattering data itself to create a three-dimensional envelope. Rigid body modeling is probably one of the more common things that you'll see done with SAXS, because it allows you to utilize high-resolution structures that you might have from other experiments and combine that with the solution scattering data to get maybe the structure of a complex or the structure of protein domains relative to each other, and allow you to do that in solution. So you can use this information, and generally the way it works is that you take, say, 2 or 3 or 4 different high-resolution structures, and you put those high-resolution structures into the modeling program, and the program will search different conformations and orientations of those molecules. And it will calculate for each one of those different models a chi-squared discrepancy between the SAXS data, the experimental data, and the model that it's currently evaluating. And it's going to minimize the discrepancy between those two to come up with your final model. And in doing so, it's essentially creating a hybrid model using the high resolution data from either crystallography or NMR, and combine that with the low resolution solution scattering data from SAXS. You want to also keep in mind that this is going to be heavily biased because it's got this high resolution information in it. So what you want to make sure you do is pretty much all journals will require you to do this anyways, is that you calculate an ab initio envelope from the raw scattering data itself, and you just want to make sure that the envelope matches pretty well to your high resolution rigid body model. So one of the programs that allows you to do this from ATSAS is called SASREF. This allows you to search for multiple different subunits, different domains to search for the complex. One of the very powerful things about it is it can actually utilize multiple scattering profiles. So if you happen to have a situation where, say, you've got 3 subunits, subunit A, B, and C, and you know the structures of each of the subunits individually, and you know the SAXS data of, say, A and B and C, and then A, B, and C all together, you can actually use all 3 of those curves simultaneously within the program, and it'll use all that information to give you the best possible model to make sure that the contribution from A and B matches the scattering from A and B, and the contribution from the B and C orientation matches the scattering of B and C, and that the entire complex matches A, B, and C. You can also, if you don't happen to have all of the structural information, say you've got missing linkers or regions with structures unknown, you can utilize programs such as Bunch and Coral, which are also from the ATSAS package, to try and get at some of that information. You can also throw in contact distances as restraints. If you happen to know this information a priori from things like fret measurements or from maybe some mutational studies, you can throw this information in and say, I know that residue 225 in subunit A is 5 angstroms away from residue 336 in subunit C. It will use that information to restrain the modeling. So docking is kind of an extension of rigid body modeling. It's actually a way of combining SAXS with protein-protein docking methods to get a more accurate, more biologically viable and energetically favorable interaction between the surfaces of your molecules. So quickly, I'll go over a brief review of what docking itself is. So docking is where you take essentially, say, 2 molecules and you roll one over the surface of the other, and you look at the energy and the surface interactions between those, and you try and sample as much of the conformational space as you can. And then you calculate for each one of those different candidates an energy score based on hydrophobicity and electrostatic interactions and surface area interactions. And you try and determine these energy scores, and then you cluster them into different groups based on these scoring functions. And you want to minimize and try and predict what the interaction is. Now, the problem with this is that there's so many different conformations that you can't search all of those in a relatively small amount of time, so they sample it with different mechanisms. Now, this is where SAXS can help, because SAXS can add in some filtering. Being able to actually determine from all of those thousands of different candidates which ones are actually the most correct ones is a very difficult task, because a lot of those energies are going to be very similar to each other, even though the conformations might be different. And so SAXS can help by allowing you to filter for gross size characteristics from, say, something like RG, which is the radius of gyration, and then take that smaller pool of candidate conformations and perform a SAXS scoring function where you're talking more fine detailed information about the SAXS data, and try and then take that smaller remaining pool of candidate conformations and continue your docking where you're clustering based on energy scores, and trying to find the interface between 2 molecules that both satisfies the SAXS data and minimizes the energy between those 2 interfaces. Some protocols allow you to do some additional conformational refinement in order to really get at what that high resolution information is. So the reason why SAXS-assisted docking is helpful is because there's some major hurdles in the typical docking protocols that don't use SAXS information. Number one is that there's such a large search space that you need to be able to sample the conformational space very efficiently. And SAXS can help because it can employ filtering in order to be able to enhance the existing filtering and scoring functions within the docking protocols. Now, SAXS only provides low resolution information, and the surfaces are not optimized in standard rigid body modeling protocols. But by combining the two approaches, you can get the optimized surface interface and the low resolution SAXS data to really get the most accurate assembly possible by combining all these different pieces of information. So one of the things that occurs in rigid body modeling is that sometimes you will never be able to find a solution. So if you take an example where you solve a crystal structure and then you just look at the solution scattering of that crystal structure, they might not match. And the reasons could be that things like crystal contacts, or maybe there's some chemical additives in solution, they might trap the molecule in some conformation that is not what its natural conformation is in the solution that you're examining in SAXS. So maybe you want to probe different chemical additives or different ligands in solution and really look at differences between the conformations from your high resolution information to your solution data. And SAXS can provide that. So flexible fitting is a way of attempting to model that information, that conformation from your solution data, by kind of morphing the high resolution model in a biologically relevant manner that allows you to fit that high resolution data into the low resolution SAXS data. This is actually commonly done in cryo-EM because you have a three-dimensional cryo-EM envelope, and they'll take high resolution structure and fit it into the cryo-EM envelopes. And now you can actually do this with SAXS data. And in fact, one of the ways to do this is by taking your SAXS bead model and actually creating a EM map, and then using that in these flexible fitting programs. One of the common ways to do flexible fitting is through normal mode analysis. So essentially the normal modes are low frequency vibrations within a protein that can be calculated, and they've turned out to be very realistic when compared with other molecular dynamic simulations. And you're essentially using normal mode analysis to generate many different conformations of molecules that are biologically realistic. So what you can do is you can fit those conformations to the Ross scattering profile or to the real space pair distribution function, or even to the three-dimensional molecular envelope. Now, I just want to throw one caveat out there. One of the problems with using a three-dimensional molecular envelope for SAXS is that in SAXS, your three-dimensional envelope is actually a model of your data. So in doing flexible fitting into a molecular envelope, you're actually modeling a model. And so there's some obvious caveats there. So generally the two main methods for doing flexible fitting is by sampling many conformations and filtering them using SAXS data. And another is actually to drive those conformations directly by looking at a trajectory along a path that minimizes the discrepancy between the model and the SAXS data. Steered molecular dynamics is also another possibility that allows you to, that has come up recently, that allows you to take an approach, but it's very computationally expensive, as you can imagine. So flexible fitting is a way at looking at some of the different conformations that you might not be able to sample in your high resolution experiments that SAXS can sample. However, it's still making the assumption that you have one single model that represents your scattering data. And sometimes that might not actually be true. You might have several different conformations that exist in solution and over the time course of your experiment, and this dynamic information is actually within the SAXS data. So one of the ways that you can try and get at this dynamic information is through ensemble modeling, and this is actually very popular lately as a modeling approach to SAXS data, because SAXS data is one of the few methods, and probably one of the more powerful methods, that can actually describe large-scale conformational dynamics in solution. Ensemble modeling works by attempting to model the SAXS data with more than just one single model, and it will average all of the models in a given ensemble to try and match your data best. So it'll actually calculate the scattering from each one of those models and then average that scattering appropriately so that you can match your data. So several different algorithms exist for ensemble modeling. The ensemble optimization method is from the ATSAS package. There's also BilboMD and the minimal ensemble search, Eros, BSS SAXS, and flexible Meccano. And these all works in slightly different ways. But the two main stages of ensemble modeling are the generation of a pool of thousands of different random conformations. And then the second step is selecting from that pool an ensemble that best matches the experimental data. Now, some of these methods do one or the other or both. And in terms of generating the pool, you can do this by either randomizing the geometry in linker residues. You can do this through normal mode analysis or even molecular dynamic simulations to generate your pool. And the selection of the ensemble generally uses a genetic algorithm to determine what that ensemble is, whose average scattering accurately represents the data. Now, one of the nice things about ensemble modeling is you can actually use this as an alternative to rigid body modeling by setting the ensemble size to 1, because essentially what it will then do is just search through that whole pool of random conformations to find what the best single conformation is from all of those thousands of different conformations. And then you can use this as a good starting point for refinement and minimize your search space for rigid body modeling if you think that you have a rigid system. So the way ensemble modeling works is you start with a pool of random conformations. I should say that one of the very useful things about SAXS data is it's one of the few methods that can actually get at the organization, the structure, and the distribution of intrinsically disordered proteins. So it's not just used for rigid molecules that have defined shape, particularly in higher level eukaryotes. It's well known that there's large intrinsically disordered regions that really have no defined structure. And SAXS can get at some of that information. So you start with a random pool of conformations. Usually the pool is somewhere in the neighborhood of 10,000 different conformations. Using a genetic algorithm, the program will select different ensembles, and then you calculate the scattering, the average scattering of those ensembles, and you compare it with your SAXS profile and you determine which ensemble best matches your experimental data. Now, in doing this, you really don't want to look at the models themselves. So most of the modeling procedures I've talked about so far, they end up with a single three-dimensional model, and you can look at that three-dimensional model and get some idea of what's happening in real space. In terms of ensemble modeling, however, you really don't want to believe the structural models that come from the final ensemble, because they're really just used as a tool to describe the gross structural properties of the ensemble. So the way that you evaluate this information from the ensemble is by actually looking at the size distribution of the ensemble, and you compare it to the size distribution of the random pool. So we have an example of that here down on the right, and there's some things that you can do. So usually one of the first things you do is you would look at how wide the distribution is. If your distribution is as wide or wider than the random pool, then that would suggest that you have a large degree of flexibility, because the random pool in and of itself obviously tends towards a highly flexible and highly dynamic population. And so if it's as wide or wider than the pool, it would suggest that it's also very flexible. By looking at where the peak position is of your ensemble, this can help you to know whether or not it's an extended conformation by comparing it with the pool, or if it's in a compact conformation. Additionally, if you have multiple distinct conformations, what you'll see is you'll have multiple peaks within your pool, or within your ensemble rather. And so by looking at these different things, you can get an idea of the gross structural properties of your ensemble. So what I've been talking about so far is polydispersity in terms of conformations within your solution. Now, I mentioned earlier that from the last talk that you really want in a SAXS experiment high quality, monodisperse, non-aggregated, non-interacting solutions. The reason for that is because several of these modeling programs require that, because they make the assumption that there is a single molecule that describes your data. In cases of ensemble modeling or mixtures where you have polydispersity, either in terms of molecular assemblies or you can have conformational polydispersity, recent mechanisms and recent methods in SAXS allow you to actually access this information in polydispersity. So ensemble modeling does polydispersity in terms of conformational polydispersity, but you can also look at mixtures of molecular assemblies. Now, generally the way this is done is if you know high resolution structure, if you know the structure of the different possible molecular assemblies, essentially what it will do is it will calculate the scattering profile of each one of those different conformations, those different assemblies, and it will determine what volume fraction you have of each one. But this requires that you have a high resolution structure of all your known assemblies. In this case, we have a dimer, and it was determined to be 63%. And we have a tetramer that was formed by the crystal packing arrangements in solution, and it was found to be about 37%. And so the combination of the 63% dimer and 37% tetramer fit better the scattering profile than either 100% dimer or 100% tetramer. Now, as I mentioned, this requires that you know the structures of all your assemblies ahead of time. But there's recently been some advances in modeling that you can use to determine the structure of some of these assemblies, even if you don't know them additionally or a priori. One of these programs is called SASREF-MX, and this is an extension of the SASREF program that I mentioned earlier, but for mixtures. And this enables you to model the polydisperse equilibria within your solution when the high resolution models are available. It can actually also estimate the volume fraction. So the way it does this is it builds up the regular rigid body model that you have, and it breaks it down into its different dissociation products, and it will attempt to look at the volume fraction of each one of those and model that so that you can actually get not only the structures of the full complex, but information about the mixture itself. I've actually done this with a few of my own data sets, and it's actually amazingly accurate at pulling out those conformational and molecular assemblies. There's also a advance in terms of ab initio modeling in looking at polydispersity, and that's through gas spore MX. And this allows you to use ab initio envelope reconstructions when polydispersity is an issue. It's similar to the way that SASREF works. It's a little bit more restricted because you don't have that high resolution information that's restraining it. So it's a little bit more restricted, but you can get at some information in terms of polydispersed mixtures. Another way to get information from mixtures is using single value decomposition. And this is a way that does not require any kind of model. It's a model independent method of estimating the number of scattering components in solution. And one of the advantages of it is that it can be used to actually deconstruct a polydispersed scattering profile into the individual scattering profiles of each of the major components. And in doing so, you can actually go ahead and get that real space information from each one of those components. So hybrid modeling is where you're actually using several different complementary methods simultaneously to look at the high resolution structure of your data. Now, pretty much all of the things that I've talked about so far are a form of hybrid modeling. But there are some methods that I haven't talked about that can utilize information from nuclear magnetic resonance and things such as the different types of NMR spectra that you can get, NOESY, TOESY. You can use residual dipolar couplings, which give you information about the relative orientation of domains in solution, of subunits in solution. You can use several different scattering methods, SAXS, SANS, WAX, and you can combine all of this, and some programs will allow this large-scale hybrid modeling. Some of the software includes Explore NIH and the Integrated Modeling Platform, just to name a couple, that allow you to do this type of large-scale molecular modeling through and able to use all of this information that you get from all these different techniques. So this is one example of that. That came out a few years ago. They were able to use, I think they used SAXS, SANS, wax, crystal structures, NMR, residual dipolar couplings, in order to get not just the structure itself, but an ensemble of the different conformations available to this large complex. So that's mostly the stuff that I wanted to talk about in terms of software. And in the last few slides here, I'm going to talk about different experimental approaches to getting at more information from SAXS. So in the last talk, I mentioned that SAXS is fundamentally a contrast method. And what that means is that it's looking at the differences between the electron density of the particle of interest and the solvent surrounding it. And so when you have mixed molecular species, you actually have different contrasts. So just to show you a quick example, if you had, say, a protein RNA complex, that would mean that you have protein, which is a generally has an electron density of around maybe 0.44 electrons per angstrom cubed, and then RNA, which has a significantly higher electron density because you've got a lot of phosphates and oxygen in there. And so the electron density between the RNA and the solvent, and the RNA and the protein and the solvent, will all be different contrasts and will contribute differently. So you can actually use this information to your advantage. And that's through contrast matching. So contrast matching is a way of utilizing these known differences in electron density to try and alter the solvent conditions to be able to match out that particle contrast. This is most commonly done in neutron scattering, but it can be done for X-rays. Now, the reason it's generally done for neutron scattering is that if you look at the scattering cross sections for X-rays and neutrons of just a few common elements, you can see the differences in the scattering cross sections. So for hydrogen and deuterium, they have the same number of electrons, and so they have the same X-ray scattering cross section, because X-ray cross section depends on the square of the number of electrons. Whereas neutron scattering is substantially different between hydrogen and deuterium, because neutrons scatter based on the nucleus and the strong force. So by looking at the different electron scattering cross sections, you can see how some of these different elements scatter. And you notice that there's this trend that it goes as the square of the number of electrons, whereas neutrons, there isn't that same trend. And so you can take advantage of the fact that a nucleus, changing the nucleus, does not have nearly as great of an effect on the chemistry of a solution of molecules as does changing an electron configuration. You can't just replace deuterium with some large metal atom in order to get at something, because you're undoubtedly going to change the chemistry of what's going on. Whereas neutrons, a lot of times you can change from hydrogen to deuterium and really not see any difference in the chemical nature. You might see some dynamic difference on occasion, but generally in terms of low resolution SAXS data, you're going to see a significant difference between the neutron scattering of hydrogen than from deuterium without having a difference in the conformational state or in the protein assembly. So contrast matching is, and this is, I'm really going to talk about this more in terms of neutron scattering than X-ray scattering, but it's utilizing that large difference in the cross section, particularly between hydrogen and deuterium, in order to match out one component. So say you had a mixed molecular species of, I don't know, say RNA and protein, or protein and lipid, or something like that. You can actually deuterate one component and solvate it in D2O in order to make that component invisible, because it's just based on contrast. So if your average scattering of your protein where your hydrated protein were in a 100% D2O solution, then you would see the protein, whereas your deuterated, lipid complex or lipid component would be completely invisible in a 100% D2O solution. So essentially what you do in an experiment is you ramp up in terms of the ratio between D2O and H2O from 0% D2O to 100% D2O, and you gradually see that one component being matched out, because there's no longer a difference between the per-deuterated component and the D2O solvent around it. And so this allows you to get at this information when you have complexes. This can be very useful in cases where you want to look at, say, one component, how it changes conformation when it binds to another component. And so you can actually deuterate the one component and then contrast match out using a D2O solution and be able to look at how the other component actually changes conformation without looking at the contribution from the neighboring deuterated component. So you can actually build up different complexes, say if you had multiple subunits, you could look at a subunit up here and then a subunit down here that look to be disconnected and look at the distance between them or the conformation between them, things like that. And so this is a great way of getting at this information. It is possible to do contrast matching with X-rays too. It's a little bit more difficult, and it's applicable to a smaller range of proteins or of molecules, because you generally need to find a solution, a solvent that matches the X-ray scattering of some component in your system. So if you have, usually for proteins, they say something like a 65% sucrose solution will do it. The problem with that is that a lot of proteins don't like to be in a 65% sucrose solution. It's highly viscous and it could cause some problems. But it is possible to do contrast matching using X-rays as well. So when you're modeling this data, you can model it using a program called MONSA. There might be other programs. I'm not aware of any, but I'm sure there's probably some other programs that allow you to model the ab initio modeling of neutron scattering as well. But MONSA is really a beefed up version of DAMN. It's much slower and it's much more difficult to use than DAMN, but it is extremely powerful. It's probably one of the most powerful modeling programs in SAXS or SANS. And it allows you to model up to 4 different electron density contrasts. And that allows you to take into account mixtures of protein, RNA, DNA, and lipids. Allows you to use both X-ray and neutron scattering data. And very importantly, it allows you to use multiple different scattering profiles to simultaneously refine against. And so if you've got complexes of subunit A and subunit B, and you want to know how it attaches to B, and where B is say a membrane protein, you can actually contrast match out the detergent or the nanodisk, whatever it is you're using, or you could model the detergent itself with a different electron density, and then you can look at the complex of A and B along with just B within the membrane protein itself. And so you can throw all of this information in there simultaneously to get at what the complete picture is. And this is what makes this a very powerful package. So finally, I'm going to talk about time-resolved SAXS. And time-resolved SAXS is a great way of looking at the solution scattering, the solution properties of a molecule over a series of time steps. Now, there's 2 main approaches to doing this. One is mixing and another is pump probe. Mixing is essentially shown up here on the upper right. And what this allows you to do is you essentially take, in this case, it's an unfolded RNA that folds when it mixes with magnesium ions. And essentially you, in this case, you mix them through some microfluidic channels, and you put your X-ray pulse, your X-ray beam, at some distance along this. So here they're still mixing, and along the trajectory of this stream, they've had more and more time to diffuse and more and more time to mix, more and more time to fold. So you essentially scan it at different lengths along the stream, and that can be worked out to be different time scales. Generally, this can get down into the millisecond and even sub-millisecond time resolutions, and it's applicable to many different systems. You can look at conformational changes upon binding to some ligand. You can look at folding and unfolding processes. It's very powerful. Another approach is pump probe. Pump probe has the ability to be able to have very short time scales into the microsecond time scale, sub-microsecond time scale with synchrotrons, and even into the sub-picosecond time scale with the new X-ray free electron lasers that are coming online. But one of the problems with it is that it's got a much more limited scope of the systems that can be applied to. You need some kind of photosensitive reaction in order to take place, because the way it works is you have some kind of light pulse that passes through your sample and causes a photosensitive reaction to trigger, and then you probe that by passing your X-ray pulse through it and collecting your data. And essentially, the way you look at different time steps is by changing the time between your light pulse and your X-ray pulse, your pump and your probe. And by looking at that, you can see that as a course, as a time course over very short time scales. This is just a quick look at some of the examples of this. This was a case where they were looking, I think it's the same case where they were looking at the RNA folding that I showed in the previous slide. You can see that Rg, the size of the molecule, as a function of time, changes significantly as it's folding. And you can look at this and see that here we have a Kratky plot, where a Kratky plot that has a peak shows foldedness, and a Kratky plot that does not have a peak but just keeps plateaus at higher Q values shows that it's unfolded. And you can see as we step up through increasing amounts of time, we can see the folding process occur by mixing with that manganese or that magnesium ion. This is a very recent example of a probe experiment done at the LCLS free electron laser. And you notice that they are able to get into the picosecond and sub-picosecond time scales in order to do this. Now, this isn't quite a matured technique yet. You really, so far, there haven't been any publications that have shown that you can get the actual SAXS data. This is actually difference profiles between the known structure and the zero time point and the subsequent time points. So it's not quite a SAXS profile, but you're getting very useful information, as you can see, because they know the high resolution structure in this case. So just a quick summary, SAXS, I hope I've convinced you that SAXS is an extremely powerful technique when you combine it with other structural data. I've shown you that there are many different methods to use SAXS data to analyze your system, depending on how much information you have and what type of information you have that you can throw at it. SAXS is one of the few methods that can access large dynamic motions. And depending on your system, SAXS can provide unique information that is not available to other techniques. So with that, I'll take any questions.
All right, great. Thanks, Tom. So if anyone has any questions, feel free to chat them to me in the chat window. I can kick one off. Could you talk a little bit about how you presented a few different cases? Maybe you could just pick a few different ways that you detect or evaluate overfitting of your data. So you've got some cases where your data is, sort of, it's not as rich sometimes as other sort of three-dimensional approaches. You may have to use different metrics or different techniques to make sure you don't overfit.
Sure. So I guess a simple way is just to look at ab initio data. So ab initio data or ab initio modeling is one of the ways that are a little bit more prone to overfitting because you're only fitting the SAXS data. So sometimes with ab initio modeling, what you'll see is you might get some kind of thing that we like to call chicken legs. So you'll get a blob, and then you'll get this long tail that comes out of the blob. And the reason for that is because overfitting in SAXS data is really caused, or at least one of the causes, is that a model will fit the noise in the data rather than the actual signal in the data. And obviously that's essentially the definition of overfitting. But in terms of SAXS, this is driven by the maximum dimension of the particle, which is Dmax. And the reason for that is because the information content in a curve is directly determined by the maximum dimension in a particle. If you've got two atoms farthest apart, scattering, they have the highest frequency in your SAXS profile. So if you have a model that has an artificially extended Dmax, then it actually allows for a higher frequency in modeling the SAXS profile, which will then translate into modeling the noise rather than modeling the actual signal in the data. So if you see, in particular for ab initio envelopes, these long tails, a lot of times they're doing that because it allows the program to lower the chi-squared fit because it's overfitting the data, while really keeping the shape and the volume almost unchanged. You know, it's really not going to change the scattering profile that much because it's such a small amount of data. Another common concern for overfitting is in ensemble modeling. So where's my ensemble modeling stuff? So in ensemble modeling, sometimes what you'll get is you might get populations that have a large extended region, and it could be just a small population of the ensemble, but sometimes people might assume that that's real when it might not actually be real. It might just be because when you're modeling something with an ensemble, you're throwing at it so many different parameters that it's that much more likely to overfit. And so again, for the same reason as ab initio modeling, it's getting these long extended regions or extended models that can be used to overfit your data. And ensemble modeling is particularly sensitive to things like aggregation, because again, you don't have those restraints that you have with just direct rigid body modeling or similar things. And so because you're throwing all these parameters at it and allowing it to fit them, you're bound to get some models that are more extended than necessarily the case for what's in the signal in the SAXS profile. Does that answer your question?
Yeah, great. Thanks, Tom. With that, I think we'll wrap it up. So thank you very much for the great SAXS webinar, and